Hi guys. So in this video, I'm pleased to be able to dive deep into the from the previous topic that I talked about, which is growth stimulation. And I'm going to be diving deep into the world of microRNA. So in this video, I will first explain what microRNA is, how it relates, and give an example of how it works in relation to MYC, in relation to neuroblastoma, and also reference a paper that I had the honor of speaking to the author of this paper in person at a conference where he described his work to me. And so I'll be describing some of the findings he has found in his lab and put that into the context of the idea of microRNAs. So what is a microRNA? So microRNAs are transcribed from genes, but they're never translated into proteins. Instead, their role is to regulate the translation of normal mRNA, messenger RNA. So what happens? So you have your microRNA genome, which has transcription via RNA polymerase to form a primary microRNA. This is then cleaved inside the nuclear membrane, inside the nuclear envelope, to form your precursor microRNA. This is then exported out through the nuclear membrane into the cytoplasm, where it is then cleaved by a molecule called dicer, to make your mature RNA. Mature RNA then has the effector function. I believe it's is it ligase helicase, some kind of microRNA helicase then basically breaks the bonds between the two microRNA strands to make individual strands. The individual strands then have the final effector function. microRNA can then have one of two activities. It can either directly or perfectly bind to the messenger RNA itself, leading to its degradation. This requires, a very specific biochemical interaction, so they need to be almost identically complementary to each other. If it's not fully complementary, then it can at least bind onto the ribosome and basically prevent full translation of a protein of a messenger RNA at the level of the ribosome. The complex which causes these breakdowns are called the microRNA RNA-induced silencing complex, RISC. So now here's the ultimate principle. microRNAs are the regulators of messenger RNAs. Messenger RNAs are the effector molecules of your genes, and these genes could be oncogenic, such as tumor suppressors and proto-oncogenes. Now we will talk about MYC and neuroblastoma. So, in neuroblastoma, we know that 25% of malignant neuroblastomas, they have an N-MYC amplification. Why is this relevant? Well, you have your growth signals, which then stimulate RAS, which then, well actually no, let me forget RAS actually, that's a mistake. You have TGF-beta, transforming growth factor beta, which is very much associated with like mesenchymal cells and repair mechanisms, activates SMADs, which then can activate MYC. N-MYC amplification is associated with the upregulation or downregulation of certain microRNAs that then result in the aberrant cell cycle. I will explain the pathology of MYC in the next slide. For now, let me explain the mechanism why a 1p36 deletion is associated with a poorer prognosis in neuroblastoma. The reason for that is because 1p36 deletion reduces the expression of microRNA 34A, which is a potent tumor suppressor. So coming back to this original concept, your microRNA 34A is primary first, then precursor, then mature, then it forms the RNA-induced silencing complex, and then this will basically break down my messenger RNAs which are associated with oncogenesis. If you delete this, you lose this tumor suppressor, which leads to feeding into the cell cycle and oncogenesis. Now let me talk about a novel finding which required discussion with the original author at a conference recently. So this individual and his team in Moscow found that lack of microRNA 128A expression is associated with a let's have a look. Poorer prognosis. Okay, so let me explain now. So you can think of microRNA 128A as a bit like microRNA 34A. It is also a tumor suppressor microRNA. And the lack of it then results in loss of tumor suppressor activity, which then increases the upregulation of a molecule called TERT. Now, it's not very well known how TERT feeds into the cell cycle, but we do know that TERT is associated with poorer prognosis, as he also shows in his paper. By the way, this was a very interesting paper which required basically real patients with neuroblastoma, histological and cytogenic analysis of neuroblastomas with basically it involved analyzing the RNA and microRNA levels in the tumor cells themselves, and also using quantitative PCR to correlate the pathology with the long-term outcomes of the patients. So it was a very beautiful paper which linked pathology to long-term clinical outcomes. And he postulated that this is the interrelation between all these systems, that microRNA 128 is normally a downregulator of TERT, and when TERT then feeds into its activity via upregulating SMARCA4, which is incidentally associated also with atypical ATRTs, atypical rhabdoid teratomas in the brain, which feeds into the beta-catenin, Wnt beta-catenin pathway. Where does NMYC come into it? Well, it is thought that NMYC is involved in also down-regulating this microRNA as well. So I hope. So in summary, in this video, I have given an introduction to microRNAs, where they come from, how they form, and what their role is. I have given two examples of microRNA activity in the relevance to cancer. I used neuroblastoma as an example, hinging around NMYC. I focused on microRNA 34A as a tumor suppressor and microRNA 128A as a tumor suppressor. 34A, I don't know exactly how it acts, but it comes from reduced expression comes from a 1p36 deletion, which is a well-known prognostic factor, well established. And microRNA 128A upregulates TERT, which plays some role somewhere along the way in oncogenesis. So I hope you enjoyed this video, and I hope it provided you with more than just scientific knowledge, but also an appreciation of how basic science can be translated to clinical research as well. Thank you very much.